Welcome back to my channel Knowledge Chronicle.
Today we're ripping open the genetic toolbox. You'll understand how humans edit life itself, what genetic engineering is, the tools behind it, like CRISPR, real-world examples such as GM crops and gene therapy, and well, the scary, exciting idea of designer babies. Let's go. What is genetic engineering? Genetic engineering is the deliberate changing of an organism's DNA to give it new traits. Think of DNA as a gigantic instruction manual. Genetic engineering lets scientists edit, add, or remove specific instructions so the organism behaves differently, maybe resist diseases, produce medicines, or even grow faster. Here are some key terms. Gene, a single instruction in the manual that codes for a trait or protein. Genome, the whole instruction manual. Modification, any change to a gene or genes. Basic tools and concepts. To change DNA, we use several core tools. First, restriction enzymes. These are like molecular scissors that cut DNA at very specific sequences. For example, they let us cut out a defective gene. Next, DNA ligase. It's the glue that pastes DNA fragments together after cutting. Then we have vectors, like plasmids and viruses, which act as delivery vehicles. Plasmids are small circular DNA in bacteria used to carry new genes into cells, while viral vectors are engineered viruses that deliver genes into human cells, and these are used in many gene therapies. There's also recombinant DNA, which is DNA assembled from pieces that didn't originally belong together. When we insert a human gene into a bacterial plasmid, and the bacteria makes a human protein like insulin, that's recombinant DNA technology. CRISPR, the game changer. CRISPR-Cas9 is the fastest, cheapest, and most precise gene editing tool invented in the last decade. Here's how it works in plain language. Guide RNA finds the exact sentence in the DNA manual, and Cas9 is the scissors that cut at that sentence. The cell's repair machinery then fixes the cut, and that repair can be hijacked to disable a gene or insert a new sequence. For example, scientists used CRISPR to remove the gene that causes sickle cell disease from patient cells, then put the corrected cells back. That's called somatic editing, changes in body cells that affect only the treated person. There's an important split here. Somatic gene editing affects only the treated individual, while germline editing affects eggs, sperm, or embryos. So those changes are heritable and passed to future generations. And honestly, that's where the ethical red lines appear. Genetically modified, or GM, crops are plants whose DNA has been altered for useful traits. Let me give you a couple of key examples. First, there's Bt cotton. This crop contains a gene from the soil bacterium Bacillus thuringiensis, which makes the plant produce a natural pesticide. Because of this, farmers actually end up using fewer chemical pesticides, and in many regions, yields have gone up. Another example is golden rice. It's been engineered to produce beta-carotene, which is a vitamin A precursor, and the main goal here is to reduce vitamin A deficiency. The benefits of GM crops include things like higher yields, pest resistance, and improved nutrition. Of course, there are concerns too, such as possible ecological effects, corporate control of seeds, and the risk of genes flowing to wild relatives. So, both the benefits and the risks really have to be weighed carefully. Designer babies is a term used for using genetic edits on embryos to choose traits, which could range from disease prevention all the way to non-medical traits like height or eye color. Right now, what's actually routine and widely accepted is pre-implantation genetic diagnosis, or PGD, which lets parents screen embryos for known genetic diseases. But germline editing to enhance traits? That's technically possible, but honestly, it's a global ethical no-go. The risks include unforeseen health effects, the potential for social inequality, and even new forms of discrimination. If you ever see someone advertising designer babies as a service, you should be extremely suspicious. Regulation and international consensus are still developing, since the consequences here really affect all of humanity. Gene therapy is all about treating diseases by delivering functional genes to replace defective ones. For example, there's Luxturna, which fixes a specific type of inherited blindness using a viral vector. Then there's CAR T therapy, which engineers a patient's own immune cells to attack cancer, and it's actually saved lives in cases of aggressive blood cancers. These are real, life-saving applications. This isn't just science fiction anymore. The risks in gene editing and GM organisms include things like off-target edits, immune reactions, ecological impacts, and of course, ethical misuse. That's why regulation, transparent clinical trials, and open public debate are absolutely essential. Looking ahead, the future could bring cures for genetic diseases, climate-resilient crops, and new kinds of medicines. But only if we pair science with real responsibility. If you'd like a follow-up deep dive on how CRISPR actually finds DNA, how gene therapy trials are run, or maybe a video specifically on golden rice and its real-world impact, just let me know in the comments. And hey, your one like, share and subscribe will help us create more valuable content for you.